Welcome to this explainer. For most of the 20th century, biology textbooks had a really uncomfortable question they just refused to ask. Can a parent's experience of the environment be directly passed down to its offspring? The standard, rigid answer was always a flat no. Inheritance happens strictly through DNA, right? Everything a parent might learn about its environment dies with that parent. Well, actually, scratch that. A landmark 2026 discovery has completely shattered that assumption, revealing exactly how plants pass absolutely crucial climate memory directly into their seeds.
Okay, let's dive into this. Here's our roadmap for today. First, the climate memory mystery. Then, we'll look at the actual cellular mechanism, how it senses, regulates, and delivers. Third, why this is definitely not epigenetics. Fourth, the gene bank dilemma, and finally, breeding for climate resilience. Section 1. The Climate Memory Mystery. To really get this, we have to look back at the historical context. As 20th century genetics solidified, it cleaned up a lot of confusion by assuming all inheritance was strictly DNA-based. But that kind of ignored a massive real-world anomaly. For decades, farmers and plant breeders noticed something out in the fields that just didn't fit the textbook narrative. Seeds harvested from mother plants grown in different conditions simply didn't behave identically, even when their genetics were an exact match. Seeds from a cool autumn germinated differently than seeds from a warm one. They carried this distinct vigor or a depth of dormancy that just couldn't be explained by genes alone. The older plant breeders used to shrug and just say, "Well, mother knows best." Now, in the academic world, maternal effects was the polite scientific term for this, but it kind of sat on an uncomfortable shelf in mainstream biology. Why? Because while the observation was very real, they literally had no proven molecular mechanism for it. Without a clear pathway showing how this happened, it was treated as an interesting quirk, but not something a serious researcher could easily build a career on. That is, until very recently. That missing mechanism was finally found in a massive breakthrough published in the Proceedings of the National Academy of Sciences in early 2026. Researchers Zhao Chen and colleagues at the John Innes Center and the Earlham Institute used the model plant Arabidopsis thaliana. By leveraging some incredibly advanced spatial analysis and looking closely at individual cells, they did something truly unprecedented. They were able to literally watch a mother plant sense the temperature and physically write that climate data into her developing seeds via a hormonal message. Moving to Section 2. Mechanism: sensing, regulating, and delivering. So the crucial point is, who is actually doing the sensing here? Is the seed acting like a tomato ripening in the sun, sensing the cold itself, or is the mother curating the information? To answer this, the team used reciprocal crosses, breeding plants with and without temperature signaling mutations, and this cleanly proved that the temperature signal was being read unambiguously in the maternal tissue, not in the seed itself. The mother is the ultimate climate sensor.
It's a brilliant three-step choreography. First, sensing. As the season cools, the mother plant's tissue detects the temperature drop. Second, regulation. The mother doesn't just randomly pump out hormones, she ramps up production in a highly regulated way, specifically in the tissues directly surrounding the seeds. And finally, delivery and degradation. The hormone is physically transported from the mother directly into the developing seed, where its final concentration dictates exactly how deep that seed's dormancy is going to be.
And this brilliantly illustrates the exact biological machinery for the researchers tuning in. The hormonal signal itself is abscisic acid, or ABA, which is a classic plant hormone. But the real game changer is the control system. The maternal plant uses a gene called LHP1 as an accelerator. It literally drives the accumulation of ABA when temperatures fall. Then, once the ABA enters the seed, another gene, CYP707A2, acts as a brake by encoding an enzyme that breaks the ABA down. So both the accelerator and the brake are totally under maternal control. The mother is basically telling the seed exactly how long to wait before waking up.
Which brings us to part 3, hormone transfer, not epigenetics.
Now, what's really interesting about this slide is the huge misconception that popped up when this paper dropped. Classical epigenetics involves heritable changes, like DNA methylation, that persist across multiple generations. The information is encoded in chromatin marks, but that is absolutely not what Chen and colleagues found. They discovered a physical transfer of a small molecule, ABA, from mother to seed. This is actually much closer to mammalian maternal-fetal transfer. Like where a pregnant mammal passes antibodies or hormones directly to a fetus.
And it all really comes down to the number one generation. This is a bounded one-generation signal. The mother senses the environment, The seed receives the hormonal message, and the seed uses it for an immediate developmental decision. This finding does not suddenly vindicate sweeping claims of multi-generational inherited memories surviving across decades. No, it is a temporary, one-generation head start provided entirely by the mother.
All right, zooming out to part 4, the gene bank dilemma.
Understanding that this climate memory is bounded to a single generation completely shifts our perspective from microscopic biology to the massive macro-level implications for global agriculture. Consider a seed sitting in a massive gene bank like Svalbard. Let's say a landrace was collected back in 1985 from a super-stressed coastal environment in Bangladesh. To keep those seeds viable, gene banks have to periodically grow them out and harvest fresh ones in highly controlled, comfortable glasshouses. So the genome is perfectly conserved from 1985 to 2025. But because maternal climate memory is a one-generation hormonal signal, that original stress memory from the harsh coast, it was almost certainly wiped completely clean and replaced by the memory of a cozy glasshouse after the very first regeneration in 1995.
So what does this all mean? Well, it raises a kind of terrifying question that scientists honestly didn't even know to ask until right now. What fitness is being lost in the fade? Ancient landraces were valued because centuries of local farming selected them for very specific, very harsh environments. We always assumed all that survival fitness was locked strictly in their DNA. But what if a critical part of that survival fitness actually lived in the maternal environmental signaling, a signal we systematically erase in uniform glasshouses? What invaluable resilience have we unknowingly lost?
Finally, Part 5, Breeding for Climate Resilience.
We are dealing with a rapidly destabilizing climate. And here's the real problem for modern agriculture. Maternal signaling systems evolved for predictability. If a mother plant experiences an unseasonably cool late summer, she sends a deep, winter is coming signal to her seeds. But if the actual winter turns out mild and the spring is warm, those seeds are now dangerously miscalibrated. Their deep dormancy delays their optimal germination window. For agriculture, this mismatch isn't just a minor hiccup. It results in highly variable emergence and totally inconsistent stand establishment across entire harvests, directly threatening our global yield.
Let's move to how this builds an incredible opportunity for crop scientists. Traditionally, we've relied heavily on mutation breeding, inducing genetic variation to write stress information into the genome irreversibly. It takes years and it persists for decades. But now, we are looking at maternal hormonal priming. By deliberately controlling the exact environment where agricultural seed is produced, breeders could intentionally write stress information into the developmental hormones. This would give the very next generation an immediate, customized, one-generation adaptation to specific climate threats. It's literally a temporary shield.
The Chen et al. Paper has definitively answered a century-old biological question. Yes, a parent's experience of the environment absolutely can shape its offspring through non-genetic channels. As we face a future of completely volatile weather, the challenge for global agriculture is no longer just deciphering the genome. So the question we leave you with is this: can we master this hormonal language of seeds, deliberately priming our crops for survival before a destabilizing climate completely scrambles the message?